All right, my technology was being a little cranky, so we're going to try this again. All right, so this is all about reception. And so if you had any questions about any of the 1, 2, 3, 4, 5, 6, 7, 8, 9 learning targets about reception, then I'm going to answer them here. All right, so the first is I can differentiate between the types of ligands and explain why they bind to either a plasma membrane receptor or an intracellular receptor. So the first thing I want to do is I want to talk about what ligands are. They are signaling molecules. You'll see them sometimes called that. Also, we're going to talk about second messengers in the transduction video, and so these are technically the first messengers. There are two types. They can be small and or nonpolar, and those can cross the membrane easily, get into the cell, and so they're often going to bind to intracellular receptors. Okay, intra means within, so intracellular means within the cell or inside the cell. Okay, the other kind is if they are polar or large. So let's review really quickly the cell membrane. So the cell membrane is made of phospholipids. The inside of those phospholipids are these fatty acid tails, which are nonpolar. And they do not let anything that is polar, can't write and talk at the same time. They do not let anything that is polar cross. Okay, so if it's polar or large, if it's large, it's just going to punch a hole in the membrane. Everything's going to ooze out and the cell's going to die. So that's no good. So if it's polar or if it's large, it's going to have to bind outside the cell. So it's going to bind to a plasma membrane receptor. So the difference is if it is an intracellular, if it's small and or nonpolar, and it can cross over, then it will bind, well, that's a weird receptor, but anyway, there we go. It'll come in and it'll bind to an intracellular receptor. If it is large or polar, you're going to see this receptor actually in the membrane where the ligand binding site is on the outside. And so if, let's say it's a big one, it's going to come in and bind on the outside. It's not going to be able to enter the cell. So those are the two types of ligands and why they must bind either to an intracellular if they can get in, but if not, why they have to bind to one of those plasma membrane receptors. The effect of the ligand on a receptor, it is going to cause a conformational or shape change in the receptor. And you know, anytime you change shape, you change function. So what happens is when that ligand binds, it is going to cause a change on this end, on the intracellular or the cytoplasmic end of the receptor. And that's going to allow that receptor to then activate whatever the next molecule or thing is to start transduction. I can compare plasma membrane receptors and intracellular. They are similar in that they both bind ligands and they both activate transduction pathways that lead to a response. So that's the comparisons, the ways they're similar. I can contrast, well, the intracellular is inside the cell in the cytoplasm. Sometimes it's even in the nucleus. It's not in the membrane, whereas the plasma membranes are embedded in that membrane. Another difference is the kind of ligand they're going to bind. So this one is going to bind the small and or nonpolar, whereas this is going to bind anything that's polar or large. Another thing is that this is going to activate a transduction pathway that then leads to some other response. Generally speaking, sometimes like a ligand-gated ion channel, you'll end up with transduction and response kind of right there at the receptor. A lot of times this receptor becomes the transcription factor that is causing the response. So transduction is a lot of times really short in an intracellular receptor. I can describe the structure and function of the G protein coupled receptor. All right, so because my art skills aren't the greatest, I'm going to defer to the PowerPoint for this. And so I can't really move my head anywhere, but so the G protein coupled receptor is going to be, a couple of things. So there is something called a G protein. It is called a G protein because it has a GDP attached to it. GDP is like ATP or ADP, but with guanine instead of adenine. So GDP is guanine dinucleotide phosphate. Yeah, I had to think through a guanosine diphosphate, I guess is the better way to put it. I'm thinking of NADP+. Anyway, so guanosine diphosphate, so it has two phosphates attached to it. When it is attached to that G protein, all of those are sleeping. They're inactive, they're just chilling, whatever. When the ligand binds to the receptor is then going to help phosphorylate this GDP to make it GTP. guanosine triphosphate. When that occurs, that activates this G protein, and that G protein can then leave and go activate the next step to start that transduction pathway. So you have the G protein coupled receptor. Then you also have a G protein, which is a different thing, that is attached to a GDP that gets turned into a GTP to activate all of that. Okay, so that is the structure and function of the G protein coupled receptor. The next one is the receptor tyrosine kinase. This is actually a two-part receptor, so they are separate when they're inactive. The ligands come and bind. They each get a ligand, so two ligands will bind. When they bind, they will come together and form what's called a dimer. It's from the same root as monomer, but mono means one, di means two. So it's just a two-part molecule. They are kinases, which if you'll remember, kinase is anything that can add a phosphate group to something else to activate it. So this one on the left is going to add phosphate groups to the tyrosines on the right to activate them. So it's going to phosphorylate them to activate them. And this receptor on the right is going to phosphorylate the ones on the left. So each one is a kinase that's going to phosphorylate the other. If we activate all 6 tyrosines, it will take 6 ATPs because we need a phosphate from each from an ATP for each of those. And then the good thing about these is they can activate 6 pathways at once. It can be the same pathway or it could be 6 different pathways. So you can either get a really big response because you're getting 6 times that response, or you can get 6 different responses. So that's really cool. It allows you to coordinate those responses if you need to. So that's kind of the structure and function of a receptor tyrosine kinase. The next is our ligand-gated ion channel. This to me is probably the easiest one. So when it is inactive, it is closed. The ligand is not bound, and the ions are just chilling on one side of the membrane. When the ligand binds, that's reception. Transduction is opening that gate, so super short transduction, super easy. So the transduction is just that gate opens, and then the response is that the ions flow through and do whatever they're going to do. Sometimes they initiate another pathway, sometimes they just do something else. But anyway, so that's kind of reception, transduction, and response are super fast in this one. And so that is the structure and function of a ligand-gated ion channel. And then we look at an intracellular receptor. So this again is inside the cell. You'll notice it's not embedded in the membrane. We have our ligand that's going to cross right over that cell membrane because it's nonpolar and or small. In this case, it's nonpolar. It's testosterone, which is a lipid-based hormone. And so it's it's nonpolar. It crosses right over that membrane. It binds to the receptor that activates the receptor. That's kind of your whole transduction pathway, is that it's activated. And then in this case, this one's going to move into the nucleus. It's going to bind to the DNA and cause that DNA to be transcribed into mRNA, which then is sent out of the nucleus to be translated into a protein. And so that would be your response. And so that is the structure and function of an intracellular receptor. And the last I can statement for this is I can name the most common membrane, and that is the membrane receptor, and that is the GPCR. So the GPCR is found in a lot of your pathways in your body. It is also, because it's the most common one, a lot of diseases are involved in the GPCR, and also a lot of drugs, because there's a lot of pathways that involve that, and therefore a lot of disorders and diseases that are associated with that. Therefore, a lot of our medications are also linked to GPCRs. And so that is reception and all the different kinds of receptors in a nutshell. I hope that helps, but if you have any questions, let me know.